All right, welcome to chapter 27. This is a pretty good read. I do, however, want to acknowledge that I think that there's a lot missing from this chapter, and that includes all the variation that can occur between what I would consider the archetype of male reproductive anatomy and female reproductive anatomy. And I think that as we are going through this chapter, I just want you to think about ways that this doesn't represent everyone. And, the goal here is to get a real appreciation, introduction to the real human body. But this is a good place to start. So we will begin with sort of the overall purpose of these organs and tissues, and that is to produce germ cells, which are germ cells are eggs and sperm. The eggs, in a technical way, are actually referred to as secondary oocytes, and that just kind of it basically identifies them at their stage of meiosis. That's kind of where they've already, progressed to a certain stage, whereas sperm are kind of just like little factories, produce lots and lots of sperm all the time. But the organs that give rise to those germ cells and the plumbing that supports them and allows for them to further develop in some cases is going to be sort of the heart of this chapter. I will point out that gynecology and urology sort of do separate the two female and male reproductive systems, and urology can combine with the urinary system too. But I will say, about 20 years ago, there was a real trend in medicine to want to split out female health from male health and like have a totally different doctor trained in just, female biology versus male biology. And that trend has kind of died down, but I think as you go through this chapter, you might get an appreciation for how different they really are. The hormones are so different. I mean, there is, a lot of systemic physiology to support that idea. But I don't know, maybe it was just like a thing in the 90s. So let's kind of start with just chromosomes in general. So chromosomes are your packed DNA. Chromosomes form when you're actually going to be dividing cells, of course. And most of your chromosomes are sort of this autosomal, like it basically is for everything other than your reproductive anatomy. That last pair is actually your sex chromosome, which generally speaking will be an X or a Y. And like I said, while the genetics don't tell the full story, the generic story is that females have two copies of the X chromosome and males have an X and a Y. And depending on what genes are expressed from those combos, we can kind of divide the genders appropriately. So anyway, you've got 23 pairs of chromosomes. 22 are for things other than your reproductive system. And that last pair, the 23rd pair, is your sex chromosome. So if it's been a while since you've talked about meiosis, meiosis is where you're going to take your stem cell, which has duplicates of all of your chromosomes. It's got 23 pairs, and we're going to basically split that so that you've only got one of those chromosomes in that particular germ cell, either an egg or a sperm. And I will mention prophase 1 because prophase 1 has two important events. First, you're going to form the chromosomes, which is a process called condensation. That's kind of where they wrap the DNA around the histones and it gives it that kind of chromosome appearance, like little X's or perhaps a Y. There's also this event that occurs called synapsis, which means the homologous, or the chromosomes that have the same alleles on them or the same genes on them, line up. So the pairs of chromosomes line up, and there is this potential crossing over event that can occur. Crossing over is where the tips of the chromosomes basically switch places. So here's an example of condensation. You can see that those are actually little balls of protein with DNA wrapped around those little histone protein balls. What I find interesting about condensation is that the DNA actually wraps 2 3/4 times around one histone, which is exactly the same number as spirals in the inner ear of the cochlea. I just think that's so cool. Anyway, Now we've basically got a compacted, twisted little chromosome. And during prophase one, we may see that during synapsis, which is their alignment of the pairs, they might switch their little tips. And this is important because this can basically result in genetic recombination. And, you know, sometimes I notice that kids look a lot like one parent. Sometimes they look like a combination of both parents. I just find that really interesting. Like this crossing over event does lead to some of that mixing, but there's sometimes going to be a big crossover and sometimes maybe just a teeny amount, sometimes none. Just kind of is random, I guess. Anyway, everything after that is basically what you predict. It's going to line up at the metaphase plate in the center. They're going to split apart, going to their separate ends of the cell. And then we basically have two cells. That's the end of meiosis 1. So here's kind of what it really looks like in real life. Here's prophase 1, because you can see chromosomes. They're still kind of scattered around the cells, but doesn't it look kind of like a hot mess? Except for the fact that there is like pairing and potentially crossing over from that synapsis pairing. Anyway, metaphase is kind of cool, I think. In this picture, the chromosomes are stained purple, and the microtubules that are actually kind of pushing the chromosomes around are stained red. They almost look like little hairs. So, metaphase, they're all lined up, and then the microtubules will pull the center of each chromosome towards the pole, and then we'll see them kind of split apart. So, telophase, the cytoplasm is basically going to pinch, and we should get two separate cells. This is a scanning electron micrograph picture of what it actually looks like. You can see that these are actually like little fuzz balls with lots of surface molecules. It always kind of makes you feel like the cells are naked except for the cytoplasm, but this is actually a pretty cool three-dimensional process. Meiosis 2 is really not very special, but we're just going to take those two cells that we produced from meiosis 1 and then split them apart one more time. This is kind of where we get like our half the amount of genetic material as a result. So here is where we're starting with prophase 2. So you can tell it looks like prophase, but we started with two cells instead of one. So there's the same idea. They're basically condensing, and then they're going to align at the metaphase plate, pull to their, respective ends of the cell, and then we'll get kind of that last little compartmentalization and splitting of the cytoplasm. So that's the end of meiosis. Just remember, meiosis is a reduction division where we're trying to basically cut the genetic material of that gamete or germ cell in half. All right, so like I said, we're going to kind of start with the male reproductive system in this video. The next video will be the female reproductive system. And we're going to talk about the organ that produces those male reproductive cells, the sperm, a lot of ducts, several glands that are going to contribute various chemicals to the semen so that the sperm are actually activated and protected a little bit, and of course some supportive structures. So we'll kind of just start from the basics. So I kind of like this guy's hair. What do you think? I think it's kind of attractive. Anyway, when we talk about the external genitalia of the male reproductive system, of course, we're going to start with the contents of the scrotum, the testes, the penis, and the rest is pretty internal. But the external visible anatomy is obviously going to be the scrotum, the pouch where the two testes basically reside, the penis, and then the glans penis in an uncircumcised penis is the prepuce. So the glans is sort of the physical distal tip of the penis, and it is covered with kind of a loose foreskin. I will talk about circumcision at the end of this chapter, but when you hear that term, that's basically the removal of the prepuce. It used to be about 90% of the male population in the United States was circumcised. That's come way down now. It's actually kind of in the 60s, which I think is kind of an interesting trend. But internally, we're going to see a series of structures starting from the testis. You can see on the bottom sagittal section here, the testis is the testicle. And then the sperm will be developed inside that structure. It will travel into the epididymis, which is kind of it's kind of like a posterior hugging comma-like structure on the very posterior margin of the testis. And then the sperm will kind of eventually move into the ductus deferens. If you're old school, you could call that the vas deferens. I believe vas in German just means duct, but the modern term is the ductus deferens. And from there, we'll see that there will be several glands, the seminal vesicles, the prostate, and the bulbourethral glands. Those will all contribute secretions into the semen, where it will then travel out through the urethra, through the shaft of the penis, and then through the external urethral opening, which is a little bit of a slit. Yeah, so let's start with the scrotum. Scrotum's just a sac, it's pretty loose. There's going to be a hemispherical septum that divides the right and the left, kind of keeps the testes on one side or the other. There are a couple muscles that I want to mention. There's a dartos and I guess, oh yeah, no, it is on this slide. Sorry. The cremaster muscle is also part of this temperature regulating system. The sperm need to develop slightly cooler than body temperature because they're so actively undergoing mitosis and meiosis. Overheating them will kind of drive that process to be too fast, and the sperm can be abnormal. Just like with any chemical reaction, you overheat it and it kind of goes too fast sometimes. For whatever reason, our sperm develop optimally a little bit cooler. So, depending on your external temperature or body core temperature, these muscles can kind of contract to pull the testes closer to the core or further away from the core if the appropriate temperature is happening. There's also a structure called a pampiniform plexus. I just like to say that, pampiniform plexus. It's basically a ginormous anastomoses of veins that is a countercurrent heat exchanger. This is very similar to the countercurrent multiplier effect that we saw in the kidney, but it basically allows warm blood and cold blood to kind of come next to each other so that the blood is cooling as it enters the testes and then it kind of warms back up as it leaves. So let's kind of take a look. We've got this loose skin pouch divided right down the center. The scrotal septum is like white, dense connective tissue that keeps the right and the left testes in their appropriate hemisphere. You can see that there are a couple layers of muscle. The most superficial muscle is called the dartos muscle. That's kind of like a thin sheet. And then there's an additional muscle called the cremaster muscle. The cremaster muscle is kind of interesting. You can see that it's going to go all the way up through the pelvic floor. And as that contracts, it's really going to pull the testes upward. If we look through a deeper vision here, the deepest on the right, you can see a bunch of blood vessels. Like there's one really bright red artery, the testicular artery, and then there's a bunch of kind of, I don't know, intertwined plexuses of veins. That's the pampiniform plexus that I was talking about. And that also follows the cremaster muscle all the way up through that hole in the pelvic floor, back upward. Anyway, and then at that deep level, we can see the outer white connective tissue surrounding the testes and a little bit of the epididymis. That would be like the head of the epididymis. All right, let's start with the testes. The testes are approximately the shape of an almond in the shell. If you were one of those poor kids like me that had to like crack your own nuts at Christmas time, oh my God. Anyway, almonds are so weird to me. It's very hard for me to see an almond in the shell and not think, oh, a testes. Anyway, enough about me. Your testes are surrounded by a dense white connective tissue called the tunica albuginea. Tunica, kind of like the eye, is just a layer. And albu is like white, like the egg of, like the white part of an egg is albumin. So like albuginea means the white layer. And what I'll also mention about the tunica albuginea is it kind of creates like little like lobulations, like it creates like these little compartments inside the testes, and that just separates the coiled seminiferous tubules, which is where the sperm are actually going to be produced. So your testis is not just one big loose bag of sperm producing cells, it's kind of compartmentalized into these little triangles that are separated by little extensions of the tunica albuginea connective tissue. Now, the tunica vaginalis, I bet you didn't know. The male system has a vagina, huh? Anyway, things you learn that are super fun. The tunica vaginalis is actually part of the peritoneum. So, okay, that's crazy. Testes actually develop internally. So they're actually in an embryo forming at approximately the same position as where the ovaries would be in a female. However, because the sperm need to be cooler as they develop, the testes will start internally developing, and at approximately 7 months of gestational age, they drop down into the scrotum. So as they drop down, they are still covered, not completely regularly, but they're still mostly covered by part of that peritoneal membrane, just like the greater omentum and the lesser omentum and all that kind of business. So the tunica vaginalis is just serous membrane and just kind of gives a little bit of like separation between that membrane and then the outer structure of the muscles and the scrotum, etc. Here's how this looks. So let's kind of start at the very core here, which are the seminiferous tubules. Those are the tightly coiled, like blue, teeny tiny tubes. You can kind of see that it's surrounded by this, I don't know, tan or pinkish color connective tissue. That's the tunica albuginea. And you can see that it creates like these little separations, those little lobes kind of separate the little tangles of seminiferous tubules. Now, the seminiferous tubules all kind of coordinate and combine their secretions and enter the epididymis, which is this kind of long, larger diameter coiled structure that just kind of hugs the posterior of the testis. But look down at the bottom. At the very bottom, or the inferior margin of the epididymis, you can see that they all combine into a single tubule. That tubule is going to start to straighten out and then form the ductus deferens. The ductus deferens will then exit the scrotum up through the inguinal canal and inside the spermatic cord, basically, which is just spermatic cord is like everything moving up through there, but the ductus deferens is going to like shoot up through there, and then it's going to loop around the ureters of the bladder and then basically get all of the secretions from the accessory glands and then out through the penis. Here's what it really looks like. So I labeled these for you, but I think you guys are at a point where you need less labels. So look at this on the video. It's not going to be labeled on your slides. So on the top are the seminiferous tubules. And do you see kind of there's a little bit of white that separates different compartments? Is it a really thick lobule? No. But it is basically a little bit of connective tissue just kind of isolating different portions of the seminiferous tubules. And I will just have you note really closely that the diameter of those seminiferous tubules is very small. When we look at the epididymis, which is a larger diameter tubule, it's different. It's not gonna give rise to sperm. The epididymis is just a duct. It really is just plumbing, and it has very different features. Anyway, like I mentioned, the descent of the testes is going to occur during fetal development. If it doesn't occur, however, that's a condition called cryptorchidism. And, this can be kind of a common finding in babies that are born a little early. And usually you don't have to worry about that if they just need time to kind of develop and descend. If they're undescended, it does require surgery because if those testes are overheated, it can lead to testicular cancer, basically, and that could have all sorts of consequences later on in life. So it's important that they do descend, just because that tissue is optimal at a slightly cooler temperature. But I always like the name cryptorchidism. It doesn't sound like what it is to me. It sounds, I don't know, it sounds kind of pretty. Anyway, let's talk about spermatogenesis. So genesis is the generation of, and we're actually going to be producing spermatids at this point. So when we're looking for the site of spermatogenesis, that is the seminiferous tubules, and that's important because the histology of the seminiferous tubules is going to be like a very active cellular, like metabolizing, meiotic, and even mitotic environment. There's going to be four different kind of things to look for. Well, maybe five, depending on how you look at it. The first is going to be the stem cells. The stem cells are the spermatogonia. So the spermatogonia are actually mother stem cells that can basically undergo mitosis. They just completely replicate a copy of themselves, and then that copy undergoes meiosis. So at each of those meiosis 1 and meiosis 2 events, we'll see that as a result we'll have a primary spermatocyte or a secondary spermatocyte. And remember, eventually we're going to try to make a sperm here, which has a very kind of, I don't know, sperm-like morphology. It's got a little kind of sharp head. It's got a little fat mid-piece loaded with mitochondria to power the flagella, and then of course a flagella for locomotion. So it's going to need to get smaller and smaller and more sperm-like as we go. So the spermatogonia are really just these little fat circular stem cells around the edge of the seminiferous tubules. And then the spermatocytes get smaller and smaller because the amount of DNA is going to get cut in half, right? And then they're going to start to be differentiated into spermatids. Now, I'm not going to call these spermatozoa quite yet because the spermatids are still attached and they're not actually capable of moving or swimming or nowhere near capable of fertilization. Now, there are some cells in between the seminiferous tubules. Those are called Leydig cells. And if it helps you, call these the interstitial cells of Leydig. That's kind of how I have always thought of them because it tells you where to find them. Those are the ones that produce testosterone. Okay, so here's a crappy slice. I'm not gonna lie. There's, 1, 2, 3, 4, 5, 6 cross sections of a seminiferous tubule. But do you see those kind of triangular lumps of tissue in between? Those are the interstitial cells of Leydig. Those are the ones that produce the testosterone, which drives this process. So this is just, I think, a good slice for those, but a terrible slice for the seminiferous tubules. Before we look at seminiferous tubules, I just kind of want to remind you that the spermatogonia are going to be the stem cells. Those are going to be near the very outer edge. Those undergo mitosis. The daughter cell of the spermatogonia will be promoted as a primary spermatocyte, and this still has, all of the DNA. It hasn't divided in half yet. When it undergoes meiosis, though, it forms a secondary spermatocyte, and the N just refers to the fact now that it has half of the amount of DNA. So we've split the chromosomes in half. And then we'll see that the spermatids become more and more sperm-like as they complete their differentiation process. One other important cell to note is a Sertoli cell. The Sertoli cell in the olden days used to be called a nurse cell, and I think that's a really appropriate description for these cells. The Sertoli cells kind of facilitate the changes that need to happen in the spermatid, like the ejection of unnecessary organelles, the shaping of the head and the mid piece. They just kind of drive the shaping process. So they're not associated with becoming sperm, but they kind of help the sperm out. All right, so let's look at the picture on the right, the cross section of a seminiferous tubule. You can see there's kind of dark cells all the way around the ring, and then there's some kind of, larger circles and smaller circles, and then basically the sperm are pretty invisible because they're actually quite small. But every once in a while you'll see their flagella kind of hanging out in the lumen of the cell. That can be a good indicator that they're there. The flow chart, I guess it's not really a picture. The flow chart on the left is the process of the genetic changes that occur in the ring of the seminiferous tubules. So you'll have the spermatogonium undergoing mitosis, and then we have a primary spermatocyte which undergoes meiosis. And then depending on what stage of meiosis you're in, you will see that those cells kind of shrink in half, and then they need to just get basically modified into spermatids before we can actually identify them as sperm. So here's a good example. This is a perfect cross-section of a seminiferous tubule. And when I look for spermatogonia, I look for condensation of chromosomes. I basically look for prophase. It's just really obvious to me when I see little chromosomes. And you can actually see some cells that are in prophase around that edge. Now, there's just a ton of little circles in the middle of this tubule. Those are all going to be primary and secondary spermatocytes. It's actually very difficult to tell the difference with only a week to do this chapter, so don't sweat it. But I do kind of want you to know the theory. And then I can actually see little triangular heads of spermatids where they're still attached to the Sertoli cell, but their little sperm flagella are kind of dangling into the lumen, making it a little bit blurry. So here's a much more clear vision, but I kind of like to start with a full cross-section. So there's two kind of center seminiferous tubules here. And like I said, look for the ones that are in prophase. Those condensed spermatogonia are really obvious, and they are always going to be around the edge of the tubule. Now, the ones with a circular nucleus in the middle are just going to be spermatocytes for us. Just you do know that there are primary and secondary from the theory, but they are just spermatocytes for us histologically. And the spermatids have that really kind of dark head here, and occasionally you'll see the tails. Like, it just depends on the slice and the way that they're oriented. Now, there's one more cell I want to point out. I've labeled the Sertoli cells at the bottom, and I want you to follow those lines. Those fried egg looking cells are the Sertoli cells, and you never really see the full shape of the Sertoli cell, but you can see their nucleus. And what's extremely prominent about a Sertoli cell is the nucleolus, that little dot inside the nucleus, which is the site of ribosome assembly, gives these kind of a fried egg appearance. So once you know what to look for with Sertoli cells, you see them all over the place. But they don't have a lot of edge that you see. So look for those too. All right, just some vocabulary that's kind of frustrating. Spermiogenesis is the last stage of spermatogenesis. So remember, spermatogenesis is basically a spermatid, right? And that's not a sperm. A spermatozoa is a sperm. So we kind of have these different stages where spermiogenesis is that last stage where the spermatid is now a sperm or a spermatozoa. I don't think most people call it spermatozoa in the real world. They just refer to it as sperm. But by the time it's sperm, it kind of looks like a sperm, like what you expect, like a little swimming head full of, DNA. Spermiation is the release from the Sertoli cell. So I will point out that this sperm is not ready yet. It has not really finished its last little developmental processes. But spermiation is technically just the release where it can move now towards the epididymis, which is actually the place where it finishes its development. In terms of sperm morphology, this is really just a very motile structure for moving genetic information. There's a couple things I'll point out. The head of the sperm, it includes an acrosome, which is basically a vesicle of digestive enzymes. This will play a very important role in the next and final chapter, which is development. It is how it basically digests a hole through the egg so that it can deliver the paternal DNA. The mid piece of a human sperm is really just mitochondria to power the flagella. So it's going to manufacture ATP within the sperm, and then the tail is just a pure flagella. So here you go. You've got the nucleus with the DNA, and then the acrosome It's got a little teeny tiny bit of cytoplasm with just a ton of digestive enzymes. And then we got the midpiece and then the flagellum. And this is the only flagella that we see in the human body, which is kind of interesting. But that's basically just a little helicopter for, swimming pretty fast. All right. Whew, the fun stuff is the hormone stuff, right? So we will see that there is a certain amount of control of spermatogenesis. This doesn't usually get turned on very much until puberty, which is when the inhibition of all of these reproductive hormones is going to turn on. The two major ones are going to be FSH and LH. FSH is follicular stimulating hormone. LH is luteinizing hormone. These will also be seen in the female reproductive system. So it's kind of nice that they're common. But I will say luteinizing hormone is primarily going to stimulate the testosterone from the Leydig cells to be released. And there's a little conversion that happens with testosterone into dihydrotestosterone, DHT. Most people just call it DHT. But it actually makes testosterone a lot more potent. And I will point out that a lack of this enzyme, you might not have enough testosterone to actually go through the puberty and the sexual secondary characteristics that occur with puberty. It's just everybody says testosterone, but the real testosterone gets converted into a super testosterone. That's the DHT. Anyway, FSH, if you think about those seminiferous tubules as being follicles, which I hope you do, because a follicle is just a ring of cells, right? Follicular stimulating hormone is going to stimulate the spermatogonia to do their thing. And of course, testosterone is going to be involved in all this stuff. But there's two hormones here, LH and FSH, and they both kind of have a slightly different part of the story. Okay, so like I said, testosterone has got to convert into that DHT, and that is going to be an anabolic steroid, full on. Now, the prenatal effect, so this does turn on in utero, and it drives the formation of the primary anatomical structures. That would be the testes, the plumbing, the glands, the penis, the scrotum, etc. But it kind of turns off after birth. So I don't know if you've ever known a newborn baby boy, but the testicles and everything is kind of swollen because this is really driving in utero. And then it kind of stops. It just kind of goes on pause until puberty, which is like where I said the inhibition of the system is removed. And then we kind of get that last driver for the secondary sexual characteristics. And that, this is probably not new knowledge, but that's going to conclude the development of the anatomy, and then there'll be engagement of sexual urges, libido, hypertrophy of other tissues. All of that stuff's going to happen. Testosterone is a classic negative feedback system. High testosterone is basically going to be detected in the hypothalamus, and then it's going to slow down the release of the gonadotropin releasing hormone. If you remember, that is like just the general gonad stimulating releasing hormone from the hypothalamus. That is going to lessen the stimulation from the pituitary gland to release the LH and the FSH. And without that LH and FSH, we should just kind of not stimulate that testicular tissue as much. So the Leydig cells are not going to produce as much testosterone. The follicular stimulating hormone means there's going to be less mitosis and meiosis happening. It just kind of it waxes and wanes as it needs. Now, there's another kind of cool hormone here called inhibin, and I would hope the name inhibin kind of tells you what it's going to do. It's going to inhibit, right? So this is a built-in timer, and this is really important because when you're producing a lot of one type of cell, like a lot of sperm in this case, we don't want that production to be so fast that the sperm don't finish their morphological adjustments and differentiation. We don't want two flagella, we don't want two heads, we don't want, weird things happening. This hormone basically regulates that timing. So if the production is going too fast, if the cells are producing a lot of sperm, they produce more inhibin with it. And that basically can be feedback to slow things down. If sperm production isn't happening very fast, then there's less inhibin actually being produced. So with less inhibin, then everything can kind of speed back up. And usually this works pretty well. But there is, in addition to testosterone, this kind of internal autoregulatory timer, which is the inhibin. Now we got to flow here. We got to go from the seminiferous tubules through kind of a series of ducts into the epididymis and then eventually into the ductus deferens. We're going to start with the epididymis, which, like I said, is kind of like a comma, and it just hugs the posterior margin of the testes. It has a head, a body, and a tail. The head is at the top, the body is kind of along the side of it, and the tail is sort of the inferior tip. And this is where all the tubes are getting combined into one. It's a pretty long tube, the epididymis, but eventually it's going to just converge into the ductus deferens or the vas deferens. So just take a peek. We're going to start from the seminiferous tubes. They're going to go through this like area where the tubes do combine, but there's more than one. Those are the rete testes. And then these efferent ducts are basically leaving the seminiferous tubes and the testes into the epididymis. And now all the sperm from all the different areas of the seminiferous tubules are combined. So this is still pretty coiled, but by the time we reach the tail, it's starting to straighten out a little bit, and it is going to change and transform into the ductus deferens. Now, sperm need to kind of stay here for a while in the epididymis to finish, their morphological changes. They need to kind of mature, is what I'll say. But there's not new cells that are being added to the sperm. They're just doing their own thing inside of a tube. So remember that, because the histology will support that function. This is just a site for the sperm to hang before they're propelled via peristalsis onward. They're not really activated for swimming at this point. They're just kind of carried in a current of fluid and propulsion through these tubes of pseudostratified ciliated columnar epithelium. This is another example of that pseudostratified, the way we saw it in the respiratory system, but without mucus, and lots of smooth muscle. So here's kind of two cross sections of epididymis, a really nice ring in the center. You can see that it's quite tall, pseudostratified, ciliated, and all of the stuff in the lumen is basically sperm. I will also point out that there is a ton of smooth muscle in the muscularis layer of the structure, which is going to just kind of make sure that the forward motion is propulsing that sperm onward, but it has kind of a long way to go through that 20 feet of coil. Here's another really beautiful version. You can see the sperm in the center of the lumen, really nice example of pseudostratified ciliated columnar and lots of smooth muscle around it. Now, the ductus deferens is actually kind of a long tube. It's going to kind of start where the epididymis leaves off, and it's going to go up through the spermatic cord. It's going to pass through the inguinal ligament. That's kind of the base of the pelvic floor with all the muscles, and there's just a little hole for the spermatic cord to enter all the structures like the vessels and the lymph and the artery and the ductus deferens. There's a nerve too. It's going to go on the back surface of the bladder, around, loops over the ureters, and then it's going to start to interface with the glands. So we will see that the ductus deferens is going to transition into the urethra once we basically start with the prostate, but we'll get there in a second. The histology is still pseudostratified at the beginning. It might kind of thin out to be more columnar depending on how far you get, but there's still a lot of smooth muscle here that's going to help kind of the peristalsis move forward. So here's a cross section of the ductus deferens. This is actually a cross section of the entire spermatic cord. Around the ductus deferens, you can see there's quite a bit of adipose. There's tons of veins. All those little red blood cells are part of the pampiniform plexus. And then what you'll notice is just a perfect circle of smooth muscle of the muscularis. This is very active and in need of peristalsis. There's a few rugae, a few wrinkles of the epithelium of the ductus deferens, but the diameter of this tube is actually pretty big. So much wider now than even what we saw in the epididymis, and there's quite a mass of sperm inside the lumen. Now here's a really good close-up of that muscularis. You can see there's a really nice ring of the inner circular and a cross-section of the outer longitudinal. So there's nothing special here about the muscularis. It's still two perpendicular layers, but it is quite muscular. A vasectomy, by the way, is basically a procedure where we will go in and cut and cauterize a small portion of the ductus deferens. Basically, a vasectomy just makes a dead end for the sperm at this point so that it can't get any further along. It is a pretty definitive sterilization procedure. I would say that in some cases, if it's regrettable, it can be reversible, but it isn't always. So, I mean, there's like some celebrities like Dr. Phil, who's not even a doctor, but whatever. He had his reversed and then had another kid, but it's actually kind of hard to do. So this is actually a pretty permanent, what I would consider as permanent as it can be, sterilization procedure for males without tons of side effects. I mean, you still have all of the accessory glands, you still have testes, they're still producing testosterone, they're still producing sperm. They just can't move anywhere, so they just degenerate and then boom, done. All right, so here's a list of all the structures that we would expect to find in the spermatic cord. Artery, the pampiniform plexus of veins, there's definitely nerves, lymphatic vessels, the ductus deferens, and of course that cremaster muscle, which kind of just hugs everything from this point. I will point out that the inguinal canal, as it goes through the pelvic floor, does present an anatomical weakness. The problem with having a hole in the muscles here for things to move up and down is that they can be weaker, they can tear. Like, the problem with this particular area is that lifting with your back can cause exertion, which can actually tear or cause these muscles to enter that whole spermatic cord area. And if that happens, you can have intestines that fall down in there or pinched off between muscles that shouldn't have that tear. Hernias can often present as pain in one or both of the testicles. So it's kind of a referred pain actually, but like that can be the presentation that something is wrong. And it's pretty easily surgically repaired. Sometimes we'll actually put mesh and things in there. I don't know. Some of the mesh has had issues. But hernia repairs, those pay hospital bills, man. I think Adventist Hospital does more hernia repairs than anyone else on the block. I just think that's really interesting. Anyway, because this opening is there, this presents a weakness, and that is why these inguinal hernias are much more common in males. All right, just want to point out something that's kind of weird. It catches people off guard. The ejaculatory duct, it sounds like it should be really big, right? Like, whoa. It's not. It's just a teeny tiny little amount of the duct kind of in between two glands. Let's take a look at it. So let's kind of start where we left off. Let's start with the testes, and then we'll enter the epididymis, kind of the brown structure on the posterior margin, up through the ductus deferens. You see how it loops all the way around the ureter, and then it's going to interface with our first highlighted gland. I highlighted them in blue. The seminal vesicle is going to join its secretions with the ductus deferens, and now that we have our first accessory gland, the tube itself is no longer called the ductus deferens. It's actually called the ejaculatory duct. That ejaculatory duct is approximately 1 inch before it combines with the secretions of the prostate. The prostate is kind of that little roundish gland right underneath the urinary bladder. Now it has a new name. It's the prostatic urethra. And it's that for as long as it is in the prostate. But you can actually see the pelvic floor muscles of the perineum. Now, when it enters that muscular pelvic floor area, it is known as the membranous urethra. That's really short. And then once it enters the base of the penis, it is the penile urethra. Sometimes that is also referred to as the spongy penile urethra, but once it's in the penis, it's totally its own part of the urethra, and it's going to stay the spongy penile urethra until it exits the urethral slit. So our three glands here, the prostate, the seminal vesicle, and the bulbourethral glands, are next on our list. So we'll start with the first one, the seminal vesicle. And remember, the ductus deferens is going to converge with the duct from the seminal vesicle and then become the ejaculatory duct. So the contents that have been added to the sperm are basically this complex alkaline viscous fluid. It needs to be alkaline because if the sperm enter the acidity of the vagina, they would be destroyed. We want to actually neutralize that acid with some of the components of the semen. So that's kind of the first function. It's also got some sugar for the mitochondria so that it can undergo, respiration. There's prostaglandins, which kind of jumpstarts the sperm a little bit, and some coagulatory proteins. So one of the interesting things about an ejaculate, which is the sperm and the fluid that is ejaculated, is it will temporarily coagulate or clot, and then it will reliquefy. It's just kind of one of those weird kind of processes that we're going to go through. So the seminal vesicles going to have some alkalinity to neutralize any acidity in the environment. It's going to have some sugar and food for the sperm mitochondria, some prostaglandins, which is just going to be like a little shot of like energy and like viability, just kind of stimulates it, and then some clotting proteins. Here's what it looks like. It's actually really beautiful. If you look at it close up, what I'll point out about the seminal vesicle is there's actually little smooth muscle rivulets in every one of those little processes. It's highly anastomosed, which means all of those little structures kind of come together and apart. But the key is actually the smooth muscle is everywhere. Prostate is next on our list, and it's about the size of a chestnut with its shell on. It's right underneath the bladder, so prostate produces some chemicals that are slightly acidic. They're going to be full of citric acid. That's why it's a little bit of acidic. We know that citric acid can enter the citric acid cycle for the mitochondria in the middle of the sperm. There's some proteolytic enzymes which are going to do the opposite as what we just put in with the seminal vesicles. Once that semen clots, the prostatic enzymes are going to start to break it down. It takes about like 10-15 minutes, I think. So it's reliquefied, and then everything can just go forward. And then acid phosphatase, which I'm sure it has a function, but I guess I still got to read papers just like you guys. Anyway, the next and last gland for the accessory glands is the bulbourethral gland. If you're old school, you could call this the Cowper's gland. That's cool. It's about the size of a pea, and it is embedded inside the muscle of the pelvic floor. So this produces some substances that basically protects the penis. So as the semen with all of the citric acid and different proteolytic enzymes and stuff is moving through the spongy urethra of the penis, we don't want to damage those epithelial cells. So this is going to lubricate and just kind of protect the exit strategy, let's say. Overall, I would say semen is a mixture of lots of different things. It's overall slightly alkaline when you add up all the pHs. It does have a lot of proteins and stuff, which gives it kind of a milky, kind of thick, viscous texture. There's lots of interesting things in there. The typical ejaculate is several mils of semen. And if you're going to do a fertility analysis, we would want to look and actually count the number of sperm per milliliter of semen. To be considered fertile, you want to be somewhere in the range of 50 to 150 million sperm per milliliter, which is like, wow, crazy. That's a lot. And like I said, it does go through this really interesting stage of coagulation, and then it will release, and then the sperm can move forward. We think that there's an advantage in producing a plug to just kind of keep the semen in place, like either at the conclusion of sexual intercourse or maybe a little bit after. There's really easy fertility tests, and then there's really hard ones. A really easy one is to just look at the ejaculate and count the number of sperm. Like, that could be a fun lab job, right? Just counting sperm. Anyway, and then, ta-da, we have the penis, which is the last structure for the movement of the semen. But this is also shared with the urinary system in this case, so for urine too. We're going to see the penis in kind of three different areas. The base, which is the bulb, and the crus, which is sort of like the little anchor points. The body, which is the shaft of the penis, and then an enlarged distal tip, which is the glans penis. The glans penis, again, is covered by the foreskin or the prepuce. There are three columns of erectile tissue that are full of venous sinuses. I'll remind you that a sinus is the leakiest of all blood vessels. And what's unique about this particular system is as those blood sinuses engorge with blood during an erection, which actually has a term to describe it, it is tumescence of the penis, the blood shuts off the anatomical exit, and then the penis stays erect. So here's kind of your textbook picture. I'll start with the cross section or the transverse view of the penis on the top right. You can see that there are two large columns of erectile tissue. Those are the corpora cavernosa. There's also at the bottom, surrounding the urethra, which in my opinion in this picture is too round. It should be more of a like a linear slit. That is also surrounded by this kind of spongy looking erectile tissue. That is the corpus spongiosum. So depending on where you're slicing that penis, this is kind of just in the middle of the body, that corpus spongiosum can actually be larger because the glans penis is all corpus spongiosum. So this is just kind of like a typical cross section. But I do want you to know that there are these two dorsal columns, the corpora cavernosa, and then at the bottom, surrounding the urethra, or enlarged distally at the tip as the glans, is the corpus spongiosum. All three of those columns of tissue produce that engorgement and enlargement / erection of the penis. Speaking of erection of the penis, this is all facilitated through the same neurotransmitter as we've been studying this term, which is nitric oxide. So remember, NO is a vasodilator. That's going to basically allow the blood to come in, compress the venous exit so that it's trapped. And this is a parasympathetic reflex. So remember, this is like rest and digest. You don't want to be too excited. Ejaculation is sort of the conclusion of this erection process usually, and that is actually a sympathetic reflex, so that's a little bit different, but we're going to see muscular contraction, lots of peristalsis, kind of moving the fluids through the last of the ducts, through the different glands and stuff, and then there's some additional muscles that are part of this system to kind of aid in ejaculation. So I have mentioned the glans penis is covered by that foreskin, and we're seeing a real trend down from circumcision. I think it's really interesting. I know a lot of people have very strong opinions about female genital surgical procedures, aka mutilation, where they remove like the clitoris or other even more barbaric procedures that might remove more anatomy. But there's actually sort of an interesting argument to be made about circumcision. Usually this is done really very quickly after birth. I gotta tell you, there's a urologist I really have a lot of respect for in Portland, and I mean, he's like the surgeon that you want for any kind of urology procedure. And 30% of his practice is like revising circumcisions. I had no idea that they had so many problems. Like either there's like scar tissue that's occluding the urethra, or, you know, there's just a ton of things that can go wrong. And I had no idea. I thought this was like a very simple procedure. And normally in class, I think we would have a discussion about this, but I'd like you to think about it because, you know, circumcision, there has been these kind of mixed like scientific guides about the purpose of circumcision, or does it make it more hygienic or whatever. And I think you can pretty much debunk that argument except for the fact that there are some cells in the prepuce that can be susceptible to HIV. So circumcision can, reduce the likeliness of someone catching HIV from an unprotected sexual event. Um, but I think anything done on babies is somewhat controversial from an ethical perspective, and I think a lot of times it's more of just like, well, this was done to me, so I'm going to do it to my kid or something like that. And I don't know if that's maybe a healthy way to look about it, but like, anyway, you should know that circumcision is a little controversial, and the trends are really pointed down in terms of like how common it is. And there's a lot of cultural, there's a lot of religious significance that can occur, and that's not my place. But from an anatomical point of view, I will question that thought that this is for safety and I don't know, just like cleanliness or whatever. Anyway, see you for the girl show.